Hi, let's talk about pseudo-hyperaldosteronism. What is pseudo-hyperaldosteronism in one sentence? It's a condition where the effects of aldosterone are present without aldosterone actually causing them. So it looks like primary hyperaldosteronism, where aldosterone would be very high, but aldosterone is actually low in this condition. So what are the signs of hyperaldosteronism? Most commonly, hypertension. And the classic triad would also include hypokalemia and metabolic alkalosis. Why is that? Well, we'll have a look at this in just a moment. But for now, let's answer the question, how can we get aldosterone effects without aldosterone actually being present? For that, we need to understand how aldosterone works and where in a signal pathway there can be congenital disturbances through mutations or acquired disturbances through inhibition. Let's do a brief review. How does aldosterone actually work? Aldosterone is the end player of the RAAS. And the RAAS is a system for medium to long-term blood pressure control. When kidney perfusion decreases, the hormone renin is secreted, and through several steps within the blood, aldosterone is eventually produced from the adrenal cortex. Aldosterone is a steroid hormone and lipophilic, meaning it can diffuse into cells because the membrane of cells is also made of lipids. So where and how does it work? It works in the distal tubule cells and collecting duct cells. It increases the expression of, for example, ENAC, which is a sodium channel, ROMK, a potassium channel, and the sodium-potassium ATPase pump. What does this lead to? More sodium can enter the cells from the filtrate, but in exchange more potassium accesses the renal cells into the filtrate, which later becomes the urine. This exchange of ions has to happen to maintain electroneutrality in the filtrate. So as we already stated, additionally on the basolateral side toward the blood, the sodium-potassium pump is upregulated. Sodium is pumped into the blood in exchange for potassium. So net, more sodium ends up in the blood and potassium in the urine. Sodium passively pulls chloride and water with it, and this increases blood volume and therefore blood pressure. The vascular system simply becomes fuller and the pressure rises. And why does potassium leave the body in the urine? Again, because there must be a counter ion in the urine to maintain electroneutrality. What else does aldosterone do? It increases the expression of proton pumps in intercalated type A cells, which are also found in the distal nephron. This makes the urine more acidic and the blood more alkalotic. So aldosterone is usually affecting the homeostasis of blood pH. Why am I telling you all this? In pseudo-hyperaldosteronism, the symptoms are like those of true primary hyperaldosteronism. Accordingly, arterial hypertension can develop through sodium retention. Hypokalemia, so low potassium concentration in the blood, may happen through potassium wasting, and metabolic alkalosis through proton loss due to this mechanism we discussed. So why is it called pseudo? Pseudo because the actual levels of aldosterone are suppressed, and renin is suppressed too. The RAAS is completely deactivated through feedback mechanisms. Let's look at how aldosterone actually works in detail to understand the underlying causes of today's topic. This is a kidney cell of the distal tubule. Aldosterone is a steroid hormone again, and binds to the mineralocorticoid receptor, which is found intracellularly. Together, they act as a transcription factor and change the expression, meaning the amount of channels over here. At these points, we now found the first causes of pseudo-hyperaldosteronism. Different structures can be mutated with the so-called gain-of-function mutation. So let's start at the beginning of the cascade of aldosterone. If the mineralocorticoid receptor is mutated, it can be active without aldosterone actually binding to it. This is also called an activating mutation and known as Liddle-Syndrome. A similar cause is an activating mutation again, but this time in the ENAC channel, and this is called Liddle-Syndrome. Let's move to more causes then. For this, we go one step back to the adrenal cortex. Do you know which other hormones are produced there? Besides aldosterone, precursors of sex hormones are produced, but more importantly, the so-called stress hormone cortisol is produced. What does it have to do with pseudo-hyperaldosteronism? Aldosterone and cortisol actually look very similar chemically, and in fact cortisol can also diffuse into cells, and what then happens is it can actually bind, even under normal physiological conditions, to the mineralocorticoid receptor, which is actually meant for aldosterone, and lead to an activation of it. But to prevent this, physiologically there is an enzyme that inactivates cortisol, the 11-beta hydroxysteroid dehydrogenase 2. So this enzyme prevents the excess effect of cortisol on the mineralocorticoid receptor. But why am I telling you this? This enzyme can also be mutated or be disrupted by an acquired condition, and thus cortisol can act like aldosterone even at normal blood values without aldosterone actually being elevated, because it's not inactivated. So this time the enzyme can have a genetic loss of function mutation, and this is called apparent mineralocorticoid excess, or AME for short. But furthermore, it can be acquired through consumption of licorice or licorice products such as in certain tea varieties. A thorough history can always be helpful in hypertension to detect this mechanism. Let's come to the last example of today, which is Cushing syndrome. This usually describes, regardless of cause, cortisol levels that are too high in the blood, called endogenous when they come from the inside of the body, or cortisol effects when, for example, exogenous steroids are administered. So when too much cortisol is present in Cushing syndrome, this enzyme, which we discussed right now, is functional but overwhelmed. It gets overrun by too much cortisol, and cortisol, or the exogenous steroid, now also activates the receptor for aldosterone, so the mineralocorticoid receptor, independently of aldosterone. To review, all these causes ultimately look like primary hyperaldosteronism. Hypertension through sodium and water retention may occur, hypokalemia and metabolic alkalosis through the activity of the ion channels and proton pump in the distal nephron. In true primary hyperaldosteronism, aldosterone in the blood is high and measurable, and renin is decreased through a feedback mechanism. In pseudo-hyperaldosteronism, our today's topic, both renin and aldosterone are decreased. Let's summarize today's topic. First, pseudo-hyperaldosteronism mimics primary hyperaldosteronism, but with suppressed renin and aldosterone levels. If you want to learn more about primary hyperaldosteronism or Conn syndrome, you may check out this video over here. Second, causes include activating mutations of the mineralocorticoid receptor, known as Liddle syndrome, and activating mutations of ENAC channel, known as Liddle syndrome. Third, apparent mineralocorticoid excess occurs when the enzyme which we discussed is deficient, either genetically or through licorice consumption, allowing cortisol to activate the mineralocorticoid receptor. And fourth, in Cushing syndrome, excess cortisol overwhelms this enzyme, producing similar effects. I hope this video was helpful to you. Consider subscribing, and I'll see you in the next video.